Purdue University researchers have engineered yeast microbreweries within disposable batches made of freezer paper, aluminum, and tape. Simply adding a drop of water activates the yeast to show radiation exposure as read by an electronic device. It can help hospital lab workers better track their daily radiation exposure, enabling a faster assessment of tissue damage that could lead to cancer. On a commercial level, the readout device could one day be a tablet or phone. The badge could also be adopted in the future for nuclear power plant workers and victims of nuclear disasters. Radiology workers are regularly exposed to low doses of radiation when they obtain patient imagery, such as X-rays. While protective gear largely keeps workers within a safe range of radiation exposure, absorbing a little bit is still inevitable. Radiation doses creeping above regulated guidelines pose risk for developing conditions such as cancer, cataract, skin irritation, or thyroid disease. Currently, radiology workers are required to wear badges called dosimeters on various parts of their bodies for monitoring their radiation exposure. They wear the badges for a month or two and then they send them to the company that made them. But it takes weeks for the company to read the data and send a report back to the hospital. But the newly created yeast badges give an instant reading at much lower cost. The success of the badges lies in the quick and measurable response of yeast to radiation. The higher the radiation dose, the higher the percentage of yeast cells that die. Wetting the badge activates the cells that are still alive to eat glucose and release carbon dioxide, the same fermentation process responsible for brewing beer and making bread rise. When carbon dioxide bubbles at the surface, ions also form. The concentration of these ions increases the electrical conductivity of the yeast, which can be measured by hooking up the badge to a readout system. Yeast also is known to be genetically similar to human tissue. Data from the badges can therefore inform future work on how radiation damage happens to human DNA and proteins.